Hey folks, this is the second half of chapter 12, and it kind of represents heredity in new ways. Uh, Back in the older idea that we had from chapter 10 and even earlier parts of chapter 12, it was that if somebody inherited something, whether it was a disease or not, there was always just a single dominant allele, and there's a single recessive allele. But with chapter 12.2, it reveals that there can be more than one dominant allele, and they're symbolized in different ways. Like a good example here is, here's a dominant allele, makes white, but chickens also have a dominant allele that's called uppercase W, but they put a little 1 next to it. So scientists, when they find organisms that have multiple dominant alleles, not just a single one, they give them numbers or little symbols next to the capital letter to represent that it's a second or third or however many dominant allele. In this example, white's dominant, black's dominant, and check this out. The older chapters couldn't explain this pattern. The fact that some feathers are black, some are white. And before time, in chapter 10, it would have either been black or white. I mean, you couldn't have a chicken that was both colors at the same time. So this section of chapter 12, 12.2, really just investigates what explains these patterns, because heredity is more complex than just having one single dominant allele and one single recessive allele. Look at this. Here's another example. Tay-Sachs disease is a brain disease, and if somebody gets homozygous recessive genotype, they have the disease. But what's interesting is that if they're a heterozygote, they're not a carrier. They have the disease, but just not as strong as someone without it. So a heterozygous person is going to be partly normal, partly sick. Both alleles are expressed instead of just the dominant version. So in this new chapter, it's also that dominant allele doesn't necessarily cover up a recessive allele. So in chapter 10, it wasn't that dominant covers up recessive. I mean, that's true, but the world's more complicated, and chapter 12 shows that sometimes even recessive alleles can show up right alongside dominant. All right, so let's look at what it wants you to do for this slide. It says, in your notebook, so in your notes, it says, if one color was dominant over the other, how would the chick's phenotype be? So based on the chickens, if one of these colors was dominant over the other, what would you expect the babies to look like? Now remember, in this example, there's two dominant. But this question is asking, if it was the traditional situation where one was dominant over the other, how would the chickens be? And then question number 2 is, how is the R allele in Tay-Sachs disease, which is this right side here, how is the recessive R not truly recessive? And we've already kind of discussed it. Good luck. All right, let's check. Let's get this slide. It doesn't help you. Incomplete dominance. So we said there's more than one dominant allele. Well, here we go. One example of the situation is called incomplete dominance. And it's when someone, basically, whatever two alleles they have, neither is covered up. And because neither allele is covered up, there's no such thing really as a recessive allele. So they're all shown in capital letters. Notice that this is a dominant R allele, and this is dominant R, but a little symbol is next to it to represent that that's a second dominant allele. And when you have this situation of incomplete dominance, you can have more than two possibilities for phenotype. So like if you had a red flower mate with a white flower, what do you know? You have a new phenotype, a third possibility, and this is light pink. It's like halfway between the white and the red. So the babies would look like they're halfway between the red and the white flowers of the parents. And this picture perfectly shows that. You have white flowers in the background. You have the more stronger colored kind of pink, and then you have these middle values. So this is a potential example, not necessarily, but it might be an example of incomplete dominance. In your notebook or scrap paper, you are to create a letter symbol for the flower color gene. So just like in chapter 10, when you have a trait or a gene, you pick a letter, any letter. And you're going to produce a letter symbol for each color possible. So you have 3 colors, right? So you're going to, basically, whatever letter you choose, is going to be used to symbolize this color and this one. It needs to be the same letter. It can't be different letters, remember? And they're both capital. But just like in this example, you have two dominant alleles, you're going to have the two dominant alleles here also. But one of these, you'd give the dominant plain R, and the white one, you'd give the dominant R with the asterisk next to it. Or you could give a different symbol. You could put anything here. It doesn't matter. You could put an emoji. Whatever makes you happy. All right. And then it says, determine the genotype for a pink flower. So the genotype for a pink flower is halfway between the two. So what do you think the pink flower gets? So what do you think its genotype is? Do you think it's two plain R's, two dominant R's with an asterisk, or do you think it's one of each? Remember the pink one, which is kind of halfway between the two. You decide. Think hard. All right, here we have an example of two different horses. Again, it's kind of the color of the animal. It's easy to understand, or easier. You have chestnut horse, you have a white horse. And if they mate together, they don't produce all white, and they don't produce all brown. They produce this new color called roan. Looks like it says, in this example, both allele versions for well, it says flower, but it'd be horsehair color. So ignore the flower, it should say horsehair color, are expressed. So white and brown are expressed, and so there's on this horse are individual white hairs and individual brown hairs. But when you look from a distance, it looks like this perfectly blended color. But really, on this horse, you have two different hair colors sprouting. It's the white hairs and the brown evenly mixed to make this lighter brown color called roan. All right, so explain why scientists no longer use the lowercase r recessive allele in these situations. So scientists wouldn't use or create a recessive allele or lowercase letter, in this case a lowercase r. Why is that? Why don't they use that now? So think about this question, and again, you don't have, there's no perfect answer, it's just you thinking through it. So, and when you're answering it, you'd be writing your answer for, let's see what slide it is. You'd say slide 13, and you'd write your answer and just show your thinking. So if you're writing, I don't expect a perfect answer. I just want to see your thoughts and you trying your best to explain why a scientist, given this situation, wouldn't believe in using a lowercase r, but only dominant r's for it to symbolize the alleles. All right, here's slide 14. We have Charlie Sheen and a random Google Pics model. And let's see. So instead of hair color, this situation is showing incomplete dominance, but for hair type, not hair color, but straight hair or tight curls. And we set up a 4-box Punnett square for you. I'm setting it up for you. And the dominant R represent the straight hair alleles that Charlie Sheen has. And he's quite happy with his hair. And then you have this lady, and her two alleles she has are dominant R still, but we use the number 1 just as a symbol to represent the second dominant allele in this situation. So this is her eggs, and this is his sperm, and you fill in the inside squares for the offspring. And then once you do that, once you fill in that, the questions are, for your notes, what would you expect the hair texture to look like if his dominant R allele and her dominant R1 allele were together in an offspring? What would their hair type be? Would they be straight? Would they be tight curls? Or something in between? In this case, all 4 children get one dominant plain R allele and one dominant R1 allele. It says make a Punnett square for the cross above. So if you haven't done that, please do it already. Fill in the Punnett square. And then predict or hypothesize what you expect the hair type would be. All right, good luck. Let's move on. So here's another situation. You have what's called codominance. And in codominance, you don't get a blending of the however many dominant alleles involved. They're kind of equally and separately expressed. And possible ways for us to think about this are like when you have solid sections of a puppy's fur that are all black and solid sections that are perfectly white. It's not a blending of 2 dominant alleles. So it's not a black dominant allele crossed with a dominant white allele to make a gray dog. You just get these solid patches of black representing the dominant black fur, and then you get solid patches representing the in this case it's the dominant B with a quotation mark symbol or white fur. So again, it says alleles don't blend. They form to form a middle. They just are both separately expressed. And here's a perfect example too. Look at this. This bird is showing separate red, yellow, and blue feather colors, and that's genetic. And it says, looking at this bird, use the word codominance and alleles to explain the bird's phenotype. So study the definition up here and use that to try to support your ideas of explaining what's going on with the bird based on codominance. And then create symbols for the 3 allele versions. Remember, for feather color, you have to choose just one letter. And then, just like in this example, for the dog, you had 2 versions, black or white. You notice it's still the B, the dominant B. So you need to pick a letter for this and follow the same style pattern. But in this case, the bird's a little more complex. Instead of just having 2 versions, the bird has 3. All right, let's keep moving. Here we have a situation we visited earlier in chapter 12 called Sickle-Cell Anemia. And if somebody inherits 2 of these dominant H's, they have normal hemoglobin, and that's what it looks like, the hemoglobin protein. If they inherit 2 of these dominant versions of the alleles, they will have the mutated protein, like this. But if they inherited 1 of each, the question is, because it's codominance, what would you expect? So based on patterns of codominance, if you got 1 of each, does it mean all are normal, all are sickle, or does it mean something else? Does it mean that you get a little of both? So restudy codominance to try to figure out what to expect if you took 1 person, a male here, for the Punnett up here, right? Remember this 4-box Punnett square. And then the female, who has 2 dominant H sickle versions of the allele. And what would the offspring look like? If the man had this and the female had this, what would their children be? And then see, it says, what genotype will be semi-sick with disease? What's the probability to get only sickle allele? So once you fill this out, you'll know the answers to both. The third thing is, it says, describe blood cells of a dominant H sickle individual. And as explained using the word protein. So here are the pictures of the proteins, what they look like. Here's the normal protein, and here's the sickle appearance of the protein. Okay? So when you talk about a person that has sickle cell anemia, what does it mean about the protein? It used to look like this, now it looks like this. And we have videos here that are perfect to better understand the disease sickle cell anemia. Of course, watch them, and it brings really to life this whole problem. And sickle cells affects a lot of people, particularly in Africa. So it's interesting. Please see it, and it'll probably help you understand the situation. Here we have multiple alleles. Look at this. You don't just have 2 dominant alleles. You got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Look at this. It's 3 alleles, or around 10 total alleles for a trait. And the trait is the color of fly's eyes. So it's not, you don't just get 2 possible alleles for certain traits. In this example, for fly eye color, look at all the different colors possible. In fact, scientists have found that there's up to 10 possible alleles that shape the colors of fly eyes. It says, in your notebook, lady flies love a polka dot fly. What genotype would produce this? So polka dot. So look at the alleles possible, and what should create if it was codominant, right? If it was codominant, which 2 of these would you combine to give a spotted or red and white eye color for flies? We have sex-linked traits. Sex-linked traits are any traits that are controlled by the X or the Y chromosome. And the Y chromosome is quite small compared to the X. Here you go. These are real pictures of them. And any trait that is controlled by a gene found on the X or Y chromosome is called sex-linked. Here's some simple questions. What gender is a human who inherits X and Y? What gender is someone who inherits XX chromosome? And then look right here. It says determine genotype for a man who tans well plus woman who gets sunburns. And right here, it's saying that the trait for skin tanning is found on the X chromosome, and it gives you 2 possible alleles. And it's the traditional, 1 single dominant or a single recessive. And then we have here a wonderful link that should help us better understand the sunburn allele. All right, here's another example.